All right, everybody. Welcome to another mini lesson with Miss Bear. We are today talking about proton pumps, and specifically, we are referencing the sodium-potassium pump, and we're talking about how and why ATP gets used and how it gets remade. Because if ATP is going to be used to power the sodium-potassium pump that you guys did in your stop motion videos, then powering that pump is going to mean we have to figure out how to make ATP, because when ATP is used, ADP is going to break off, as we talked about in class, and we need to figure out how to remake it. And that has to do with the following proton pump. So this is a molecule called ATP synthase. Anytime anything ends in ASE, it means it's an enzyme. And so that means it's going to be either putting a molecule together or breaking it apart. And this ATP synthase molecule is going to use a proton gradient to power the synthesis of ATP. Well, how is it going to do that? Well, I'm going to come back to this image. And I'm going to simplify this diagram, okay? And I've included here two proteins, but we're going to start just by looking at ATP synthase. So that's this guy here. And ATP synthase is going to take ADP, adenosine diphosphate, adenosine with two phosphates smushed to it. And it's actually going to smush another inorganic phosphate onto it. So that's what this P lowercase I stands for. I know it looks like a C, but it's a lowercase I. You can see the little dot above it. So these two molecules, the inorganic phosphate and the adenosine diphosphate, are going to get smushed together in ATP synthase to form ATP. Now, how is ATP synthase going to do that? Well, it's going to use a proton gradient. So I've added here the concentration gradient of protons. And so if you're looking here at this gradient, which direction would you anticipate that protons would want to move? Take a second, make a prediction. Consider where these protons would want to move based on pure diffusion. Now, here's the thing. They are ions. Even though they're small ions, they are ions. So they cannot diffuse through the cell membrane. So in order to maximize and capitalize on this proton gradient, ATP synthase is actually going to create a little funnel for those protons to move down. It's kind of like a water wheel. If you remember, or if you've ever seen in old-timey movies, they have water wheels. And water wheels are going to use the force of gravity to propel water down to power things like mill grinders and things that were used in previous generations, even fairly recently, to power different kinds of motors. ATP synthase is doing the same thing. It's maximizing and capitalizing on a gradient that already exists to power the synthesis of ATP. Now, at this point, you might recognize a problem, because as protons get pumped from the outside of the cell or outside of the membrane into the other side of the membrane, you're going to start to reach equilibrium. And as you start to reach equilibrium, ATP synthase is no longer going to function. So how do you reestablish the gradient? How do you pump protons back to the other side of the membrane? Well, in order to do that, we are going to use a molecule called a proton pump. So over on the left-hand side, the purple molecule that I've drawn is a proton pump. And this proton pump is going to take protons and it's going to pump them through the cell membrane to the other side using ATP. And it's going to use ATP as an energetic input for pumping protons against the concentration gradient. So this proton pump molecule, you've seen representations like this in your textbook. This proton pump molecule is going to take hydrogen ions, pump it against the concentration gradient, and it's going to do that using an energetic input. It's going to do that using ATP. Now, this might seem kind of confusing. Why would you use ATP to make ATP? That seems like a redundant process. Well, what ends up happening is ATP is made in way higher concentrations than it is used in proton pumping. So ATP synthase can crank out, like if we look at this GIF that I have, this is ATP synthase. You can see all these little yellow, bright yellow dots. Those are protons. And as protons get sucked into ATP synthase, the thing is going to rotate, not the thing, the protein is going to rotate. And as that protein rotates, it smushes together a bunch of ADP and phosphate. And you can see these ADP molecules, these blue ADP molecules getting sucked in, and then a purple ATP molecule being released. So this is going to be how ATP is synthesized so that we can power things like the sodium-potassium pump, which can then be used to make glucose. Hope that was helpful. And my last question that I will leave you with is a consideration of where these proteins are going to be found. If something is going to make ATP, where is it going to be found in the cell? What organelle might it be found in inside of eukaryotic cells? And hopefully you had the chance to say mitochondria. And if you didn't, go back and review your notes on organelles. Hope that was helpful. I'll see you guys later.